working?
Yep, excellent.
And can you see it advance there?
Yep, floor is yours.
Okay.
All is good.
So this, well, first I'd like to thank the organizers for giving me reason to get up early in the morning, many more reasons than I've had for quite a while. And the second is to thank the audience for still being here, at least virtually, or at least with your machines on, after an intense 5 days. So this conference about ecology and coevolution, and we're supposed to be going from models to data, and hopefully, in my case, or eventually back again, I'm only going to do a bit of motivation for the models from data and then talking about models. And I seem to have written backwards what we were supposed to be doing as well. That's not a very good start. Okay. So this work is done in collaboration with Atish Agarwala and Michael Pierce. So the basic question is, why is there so much biological diversity in all scales? And particularly, I'm going to focus on the fine scale subspecies microbial diversity, which is, to me, extremely surprising because it's often coexisting in the same physical location. I'm going to show an example. And one of the goals here is to really ask, what should one not be surprised by? If one can see some feature in simple models, then maybe one shouldn't be so surprised by that in nature. So what am I going to just give as the one example here, which is an example of a species, Prochlorococcus, dominates the photosynthesis in the mid-ocean, had been studied intensively by Penny Chisholm and collaborators. And here's some data from 96 individual bacteria from just 3 samples from one location, but over 3 seasons. And you see this complicated phylogeny that one can see. And the interesting thing about this is that the time for most common ancestors spans here all the way from thousands of years to millions of years. In this group, to hundreds of million years for all the Prochlorococcus. They mix around the ocean quite fast, and so these are globally competing. So why is it that every time one takes a sample, every cell that one looks at in it, and there's now hundreds and hundreds of individual cells in sequence, that they're all different? You know, how can we explain this? So that's the sort of basic question which we'd like to try to get at. Okay. Why is this? Sorry, I'm for some reason this is not advancing properly. Okay. So the basic question is this a consequence of the extensive diversity? Is this something which is general consequence of sufficient biological complexity? This was working fine yesterday. Okay. So in the phenotype space is very high dimensional objects. So all the properties of an organism is clearly something which is very high dimensional. And the properties of the environment are also very high dimensional. So is this a sense in which these being high dimensions enough to give one the complexities which will give rise to this diversity? Okay. So what is the basic assumption we want to make here? That's that the complexities of the biology, the ecology, and most particularly evolutionary history mean that fitness differences, how things growth rates depend on the environment, how they couple to other biology and so on, all of those are likely to be the sum of positive and negative contributions. If they were just uniformly beneficial, then any mutation would have already come in a big microbial population. Okay. So how can we characterize it? Once it's the sum of positive and negative contributions, a natural characteristic is to approximate that by randomness. And of course, this has a long tradition in physics. And this is something which we can hope that some of the results we will get out will be universal in high dimensions, at least loosely, and in some sense, which I'll talk about. Okay. So what are the basic questions? So the simplest conditions we can imagine, where there's no spatial gradients or temporal variations, everything is competing with everything else initially, even though we'll consider one step beyond that. Then we want to ask whether the populations can generically sustain extensive diversity without continual evolution. What do I mean by this? Well, this is the problem often looked at, where you assemble a community from a whole lot of different species, or in our case, just a whole lot of different closely related strains, and then you ask, will that sustain, or will a lot of them, or almost all of them, go extinct? And then the crucial question is whether such diversity, not whether it can exist in simple models, but whether it can generically evolve and persist. And then the question, which I'm going to explain what I mean and associate with dimension, which is how much complexity is enough in order to get this extensive diversity. Okay. So the most basic is one species, or also think about 2 species with very closely related subtypes. Okay, so if we want a model of the ecology of these, and this has been introduced by various people already, and apologize for the notational differences. So we have a bunch of types or strains, numbers 1 to k, with each have its population, and the basic population dynamics are the growth rate of course, proportional to n. There is a interaction, there's competition with itself, with close relatives. So this is negative self-interaction. There's interaction with other types. There's overall selective differences. I can think of these as being generalized differences. They don't depend on who else is there. And then to make things simple, I'm just keeping a Lagrange multiplier here, which keeps the total population size constant. So technically, I think this becomes the replicator equation if one wants to say it in those words. What one would like then is to ask for large K, what phases of this are possible? Phases in the sense in which things which will be precisely different have distinctions between them in the limit of large K, large number of strains. Okay. So the interactions between the strains, how can we parameterize these? So between very similar types, the first and in some ways the most crucial part is that the interaction with your siblings, the competitive interaction is not necessarily special. There's no reason generally it should be bigger than with your second cousins or your fourth cousins. Okay, and that's a crucial bit about looking at the strains within a species. And then from the sum of positive and negative effects, we're going to assume the differences between types, the interactions between those are going to be approximately random. And this follows a line talked about by Bob May and many others in between, and that both Jeff Gore and Guy Boonin referred to and talked about some in their talks. So the characteristic scale of the interactions is just set to be 1, but then the crucial bit is the symmetry parameter. So this is the relation between the way type alpha is affected by type beta and the way type beta is reflected by type alpha. Okay, so of course, if they're directly competing with each other, that's a positive correlation that would correspond to gamma being positive. But in general, the gamma can be of either sign, but in this range. So, for the most part, I'm going to ignore these selective differences. They need to be small, and I'll say something about that. That's rather important towards the end. Okay, so what is the phase diagram of this? And this has been contributed by many authors, several of them at this conference. Okay. So in terms of these two parameters, we've just got the strength of the self-interaction here, Q, and the symmetry parameter gamma here. And then there's one part which is beloved by physicists, which is this case where it's totally symmetric, and a lot of the competition models fall into that, the resource models. So that's rather special. Okay. Up here, where the Q is strong, that's a phase where there's niches. There's a unique large stable community in the limit of large K that's unique with high probability. But the crucial thing is that to get that, the self-competition has to be large. It has to be much larger than the square root of the number of strains. So what does that mean? It means if we increase the number of strains, we come down across here and we go out of that stable phase, it goes unstable and something else happens. Now, Jeff Gore said a little bit about what happens here. The existence of this was already predicted by May, but this is more general, and we really understand what happens in this whole region here. The particular line we want to look at is here, where the self-interaction is not at all special, so look as a function of gamma. Now, I'm going to mainly focus on what goes on over here, where there are anti-symmetric correlations or tendencies in the interactions. Okay. This phase diagram is changed by selective differences, but only quantitatively changed, provided they're small. Now, it turns out there's one very special point here. There's a special point over here at gamma of -1 and Q equals 0, which was going to use as sort of the base of our understanding, and it's rather surprising that might serve as the base. So how can I get anti-symmetric correlations? Well, one simple way is if I just have one-on-one competition. So if strain A beats strain B, then strain B loses to A, so the interactions between them are anti-correlated. But a more interesting way is if I have host pathogens. So I don't have a block structure, which I'm going to explain later, and I sort of have a block anti-symmetric correlations, because if, say, a phage does better against the bacteria, then that strain of bacteria will be better for that phage, but of course it's worse for that strain of bacteria. So that gives me anti-symmetric correlations. And as I'll explain this as we go along. Okay, so what happens in this special model? This is a special model where gamma is equal to -1 and Q is equal to 0. And this has a chaotic phase. It has a family of chaotic states. And in this chaotic states, roughly half of the species go extinct. But the surviving species have this chaotic dynamics, and this chaotic dynamics has wild swings on a logarithmic scale. And so this is looking at the fraction of the total population of the different strains here. If we focus on strain black, then strain black, the strains blue give rise to positive interactions on black. So I've colored those in blue. So the black will then to come up in population. It'll grow exponentially roughly here, but with variations. When it gets large, it brings up strains red here, which come up. But then those strains red will push it back down again because the antisymmetric correlations. And when it's large, it will push the blue strains back down. So I have this competition here, this goes on, I get these fluctuations up and down, say roughly half of them go extinct, the rest will persist. So that's very special. What happens if we go beyond this antisymmetric model? Okay, so if we want to stick with Q equals 0, but we could go slightly positive Q as well, but having gamma still negative, so we've still got antisymmetric correlations. So we still have these general tendencies that something will come up and something will go back down again. Okay. However, what happens is you get wilder and wilder fluctuations. The populations fluctuate wilder and wilder and bigger, and they more and more of them eventually go extinct. Okay, if you didn't have finite populations, if you're infinite population, they would keep getting wilder and wilder. But of course, then that just becomes nonsense. Okay, sometimes you get stable limit cycle with just a few of them remaining. Okay. So a crucial question is whether or not something like the chaotic phase can be stabilized. So this is traditionally done in island biogeography. And again, Jeff talked about this, of stabilization by migration. The normal is to assume there's a mainland. This assumes there's a persistent pool, and then you can recover from local extinctions on the island by getting a new invasion from the mainland, reinvasion from the mainland. However, this is cheating, because then you're just replacing the problem about the diversity on the island with the problem about diversity on the mainland. So what happens if we have instead try to do this more reasonably? So we can say I have many islands, okay? And on this possibility, I can get a local extinction here and recover from it by migration. On the other hand, I can also have global extinctions. So we want to ask, will many types survive if we're in this situation with many islands with migration between them? Well, this has been looked at in addition to by us, by Felix Roy and collaborators who have some very similar conclusions in a somewhat different regime of the phase diagram. Okay, so the idea here is that one can get, in fact, chaotic survival. Now, there's another way to do this. You don't actually need islands. You could also have spores where each type can go into can sporulate into a state which will gradually die out, be longer lived, but gradually die out. And these can then replenish the active population, but they will die out completely if they're not replenished. Okay, so they can give a feedback there. And this is related to Chesson's, what he calls a storage effect for how one can sustain some diversity. Okay. So we want to then explore this, this possibility. Okay, so this is now we're going to look at the multiple island Lotka-Volterra model. So we're going to have islands here, or demes. Okay, on each of these will be the same set of types. All the islands are identical. So we have interactions within the island are identical, determined by the same V. The migration among the islands is at some low rate relative to the basic scale of the ecologies on the island. So this is sort of spatial mean field model. And on each island, the total population will be, the total population across the whole of all the islands of type alpha is given by N alpha. So what does that mean? Well, if I have migration from each island to every other island, it means that each island will lose from migration out, but it will gain from migration coming from all the other possible islands. And so this is the average population per island of type alpha. So we just have this additional part, and we have to do then the analysis of this model. Okay. Well, this can then be done by spatiotemporal dynamical mean field theory. We can't do usual static mean field theory with replicas and so on, because this is all dynamics. We don't have a Lyapunov function. And the mean field theory turns out to be somewhat difficult to analyze. One can either do that numerically, as Roy and collaborators did, or we can do it quite a lot of things with asymptotics. So what comes out in this model? If I'm now with a negative correlation, so I've got anti-correlations in the V's, but not perfect anti-correlations. I have some negative value gamma. Okay, so what one finds is that there's a spatio-temporally chaotic phase, and a crucial part about this is that the ions will desynchronize. If there's chaos on each island and only weak coupling between them via migration, then there will be a tendency for that chaos to desynchronize. So each island has roughly independent chaos. Quite a lot of the strains will go globally extinct, but some fraction of them, of order the total number, half total number, or actually can be quite a lot more here, will coexist and persist. The abundances are spread on a logarithmic scale. There's a negative average growth rate of each type, but nevertheless, they persist of most of the types. So that's surprising, and that's coming from the fact they can persist in other islands. And a crucial part is that all the persistent strains have blooms up to large populations. So here's just an example of a strain which goes globally extinct. This strain is going globally extinct, and the crucial bit here to follow, this is the average over all the other islands. That's the total population, and that's sort of setting a floor. But if that gets too low, everything can go extinct, and this one's gone extinct completely. Okay. Now here's one where a new strain can actually come in. It comes in on one island, goes across all the islands. So here I'm showing the same strain on multiple islands. And then it can coexist, can persist over all the islands. It can have a local extinction here, but it comes in the other islands enough to bring this back. So this is what a substantial fraction of all the types will do, or in some parameter ranges, a very large fraction of all the types will do this. They will persist indefinitely, even though they can have some little local extinctions. So this is the basic property of the phase. Each one will have a bloom up on each island, and those blooms provide enough migrants in order to sustain the overall population on the other islands. Okay, so here we've shown that you don't need to hypothesize a mainland, you just need some spatial structure. Okay, so this is everything from mean field theory. This is initially done in the limit of very large number of islands, limit of very large number of strains. So how realistic is it? Well, it turns out that this phase is very robust. Particularly, we can look at finite populations per island and see the extinctions, as I've already mentioned. That'll affect how many survive globally, but it won't stop the phase still existing with many types. More importantly, we can look at a finite number of islands, and it turns out that they will survive globally for a time which is exponentially long in the number of islands, and that'll depend on the particular strain and all the parameters in the problem. So this is a very robust phase. We can expect that, in fact, it exists with real spatial structure as well. What we've done here is just where you can migrate from every island to every island, or every person's gut to every other person's gut. But we believe it should exist with real spatial structure, but that's an open direction. Okay, so far so good. But we'd now like to ask whether or not such a phase can evolve, or if it is there, if it's assembled, is it stable under evolution? And so the hardest situation to get that is with evolution is much slower than the ecology, so we can't count on the diversity being continually generated by the evolution. Okay, so if we just take the simple model, we can random anti-symmetrically correlated V's, we can add new types or existing types, and what one finds that above some number of order 50, and we don't quite know where this number comes from, if you go there, you're going to get more and more types, and this will just keep growing steadily. It'll get more and more diverse. If you mutate existing types, you can look at the function of the correlation with the parent, and you find again the same thing. That this number just keeps going up steadily. So this continual evolution and diversification, in some loose sense, they are not surprising. They can arise in simple models. Okay. However, the situation we've got here is very unsatisfactory. We don't have any phenotypes of the organisms. The organisms are only defined via their interactions with other types, perhaps with their selective differences between them overall as well. Okay. So we'd really like to ask what happens if we take the phenotypes. Okay. Oh, sorry, I should mention first, if we put in some selective differences, so we now have generalist mutations, and a lot of people have found that with generalist mutations, you actually lose diversity in simple models. That's not what happens here. It continues to diversify, but gets slower and slower. The selective differences gradually increase. The variance of those existing in the population decreases. Okay, what we would like to ask about is this question about the phenotypes, and particularly the question of how much complexity is needed in order to get this highly diverse spatio-temporally phase evolving. So the interactions we can consider via some d-dimensional phenotypes, and can we get a diversity which is only of order d, as in resource competition models, or maybe much larger than that is what we would hope. Okay. So in community resource models, where we have interaction via d resources, or more generally interaction with via D chemicals, what this gives rise to is V being a rank D matrix, and the maximum size of a stable community one can get, just an old result, is D. And in fact, if they're generally interacting via D chemicals, you'll typically get the maximum size community is actually much less than D. So we can't really go very far with that. What about if we consider what we sort of the original motivation for these antisymmetric correlations, which was where we have a bacteriophage interactions. Okay. So in this case, we have a block diagonal form for the matrix. The X's determine how the phages kill the bacteria, and the U's determine how the bacteria enable the growth of the phage. And we would expect then that to be correlations between these two types. The anti-correlation is there from the minus sign. But now we would like to make the interactions be phenotypes that are carried by the organisms. So here is a receptor on the bacteria, and X here just goes from 1 to D. You can think of that as being like crudely the amino acid sequence of the receptor. The phage interacts with this via tail, the type mu receptor. Again, this is a d-dimensional vector. Okay. The bacterial death rate, X here, is just a function of the interaction between the receptor of the bacteria and the tail of that phage. Okay, so this is now a deterministic function, and the phage population growth is a deterministic function of the same quantity. Okay, these are going to be different functions here, and we can of course allow general functions and have some biological motivation for that. Okay, so we've now got, say, interactions via these d-dimensional phenotypes. Okay, so what do we see? Well, it turns out that bacteriophage, the basic dynamics of it is very similar, as one can see from the mean field theory, to just the antisymmetric correlations. And so one finds that one gets here a spatiotemporally chaotic phase. Unfortunately, if the F and G are linear, which would be the sort of simplest thing and is often assumed in community resource models, the equivalent of that, if they're linear, then the maximum coexisting diversity is small, it's avoided D. Okay. But what happens if they're nonlinear? Well, the motivation for that would be that what matters for the bacteria is whether the phage is good enough to kill it or bad enough to kill it. And for the phage, is it good enough to get in? Of course, it can do somewhat better still if it maybe has a more optimal receptor. So a natural thing would be considered nonlinear functions here. So if one considers Tench-like functions, just generally sort of saturating functions, then it turns out, at least with some choice of functions here, is that one can evolve extensive diversity for dimensions bigger than around 5. Okay, so there's no specialization here. They're generalists. Everything interacts with everything else. Okay, if one starts with some community and assembles it, and then one can do the same thing, let that evolve. Eventually, most of the original strains will die out. The things that evolve are these properties, so these phenotypes. So what this is really continual evolution with the phage tails chasing the bacterial receptors, and both Ido Kusel and Shen-Chen Wang talked about that. So that's the kind of dynamics that comes in here. Now, we don't understand at this point how the behavior sort of depends on dimension. We do know that diversity can get extensive, way higher than D, and seems to keep growing. You can get some amount of specialization that seems to sort of come in a sort of statistical way, but generally everything is still interacting with everything else. Okay. So this is saying that maybe we shouldn't be surprised that we can get extensive diversity, even with relatively simple interactions. Okay, so for the future, what we would like to ask is, well, we first said that this extensive coexisting diversity is in some sense not surprising. We can get it in simple models. Of course, we would really like to do the other task now here, go back to connections with reality, and to have some testable predictions, or at least post-dictions, and sort of a framework. But what we've got here is a sort of scenario and a framework for trying to sort of rationalize, at least, and ask about ways in which a large number of strains within a species could coexist and spatially compete with each other. Okay. So the acknowledgements, I said, this work was done with Atish Agrawal and Michael Pierce, and Alana and Aditya and Gabriel have been working on some parts which are associated with the extensions, and also with the connections of reality, and thinking about a lot of data from the Prochlorococcus and another species, which shows a lot of diversity. So thank you.
All right, thank you, Daniel, for a great talk. We're almost out of time.